All right, welcome back to Anatomy and Physiology here with your professor, Dr. Sean Clavel. Today we're going over the sensory systems. This is the somatosensory section here where we'll talk about the senses of smell and taste. But in this further sections, we'll talk about the hearing and balance. But I encourage you, I'll kind of talk here where talk about the Remember, we've got nociceptors related to pain, thermoreceptors, photoreceptors, and what we're looking at here, both taste and smell, are what we identify here as chemoreceptors. So, chemoreceptors related to a chemical signal, whether it's an odorant, right, like smell, or something, the chemical that would diffuse over the tongue and give us the sense of taste. But I would encourage you to watch the crash course video as an introduction into the senses of smell and taste. And so the sense of taste, also referred to as gustation, is the special sense associated with the tongue. The surface of the tongue, along with the rest of the oral cavity, is lined with stratified squamous epithelium. And the raised bumps on your tongue are called papillae, or papilla for singular, and they contain the structures for this gustatory transduction. There's four types of papillae based on their appearance. There's the circumvallate, the foliate, the filiform, and the fusiform. And within the structure of the pil, pil excuse me, pillipa, uh, we have the actual taste buds, papilla. We have these actual taste buds. And it's the taste buds that contain these receptor cells for the transduction of the stimuli. So you can see here that we've got inside those little papilla, you've got the taste bud, and there's the taste hairs at the top. The information there, the chemical then kind of diffuse down and then start an action potential down towards the gustatory cell. These receptor cells are sensitive to chemicals contained within foods that are ingested and then release neurotransmitters based on the amount of the chemical in food. So, neurotransmitters from gustatory cells can activate sensory neurons in the facial, glossopharyngeal, and vagus cranial nerves. So here we have that anatomy of the tongue, the body being the kind of the bigger portion of it, where the root sits more back towards the back of the throat here. The primary location of those vellate papillae, the filiform papillae all over. But the structure that we really want to get familiar with is this of the taste pore itself. We've got the taste pore with the taste cells, or those gustatory cells, which will then send an action potential towards the sensory nerve fiber. So, as you can imagine, you're going to have some chemical that will diffuse into the taste pore that will then stimulate these cells to send a signal to the nerve. And so there's really only a few recognized submodalities when it comes to taste or gustation. And really, we only thought about four tastes until most recently when we had this taste, umami, right here at the bottom, where umami related to the kind of savory or meat-like taste. So umami is the Japanese word for delicious taste. But we might be familiar more with bitter, which is a sensitivity adaptation to like detect toxins, particularly in the back of the throat, before we have the ability to swallow it. We can gag, so that bitter taste to help protect us from toxins. Sour from spoiled food, or acidity, right? So there's going to be a large amount of hydrogen ions there that might diffuse and cause a action potential. Salt, again, there's a water or electrolyte balance that is monitored as it relates to the taste for salt or the desire for salt. Sweet, so oftentimes we associate foods that are sweet with those that have high energy, right? Carbohydrates or sugar. And then this newer one that we mentioned is umami, or protein. And so the way that these tastes work is that, as we've mentioned, the chemical will diffuse across the surface of the tongue. The will then send a signal to the receptors specific to that chemical. So in the case of salty, it's simply the perception of sodium ions in the saliva. When you eat something salty, those salt crystals dissociate into the components sodium and chloride, which dissolve into your saliva. The sodium concentration becomes high outside of those gustatory cells, creating a strong concentration gradient that drives the diffusion of the ion into the cells. The entry of sodium into these cells results in depolarization of the cell membrane and the generation of a receptor potential. Sour, similar to that, is the perception of the hydrogen concentration, right? Hydrogen ions. Just as with sodium in salty flavors, this hydrogen ion enters the cell and triggers depolarization. Sour flavors are essentially the perception of acids in our food. Increasing hydrogen ion concentrations in our saliva or lowering the pH of your saliva triggers progressively stronger graded potentials in the gustatory cells. Example might be like orange juice, right? It tastes sour because it has a pH value of approximately 3. Sweet, bitter, and umami is based on the activation of a G protein, a coupled receptor by a specific molecule. The molecule activates this receptor that activates this receptor is the amino acid L-glutamate. Therefore, umami flavor is often perceived while eating like protein-rich food. But the idea here is that I'll zoom in. Sweet, bitter, and umami bind secondary messenger signals. And so what that does is, simply stated, a secondary messenger signal will have a messenger, right? Neurotransmitter in the case here, the whatever the protein is, binding to the receptor, which causes a cascade of events inside of the cell, ultimately resulting in an occurrence of the something on the cell membrane occurring, right? The action potential moving down the cell membrane. So, as opposed to a direct connection that occurs with, like we mentioned, salty and sour, where the particular ion, whether it's the sodium ion or the hydrogen ion, binds to the receptor, causes a influx in. Or in the other case here, we have the neurotransmitter or the amino acids, again, in this case, binding to the receptor, causing a cascade of events. And so once those gustatory cells are activated, they release neurotransmitters into the dendrites of the sensory neurons. These neurons are part of the facial, glossopharyngeal, and facial and glossopharyngeal cranial nerves, as well as a component within the vagus nerve dedicated to the gag reflex. Remember that bitter taste that we've got back in the tasting, the ability to actually gag. The facial nerve connects to the taste buds in the anterior third of the tongue. The glossopharyngeal nerve connects to the taste buds in the posterior 2/3 of the tongue. And the vagus nerve connects to the taste buds in the extreme posterior of the tongue, verging on the pharynx, where the more sensitive noxious stimuli, which is more sensitive to noxious stimuli such as bitterness. So the cranial nerve 7, 9, and there's the vagus nerve number 10. All right, so the facial nerve number 7, the glossopharyngeal nerve number 9, and the vagus nerve number 10. Ultimately, the destinations of the gustatory pathway is the thalamus, right? The gustatory cortex via the thalamus. So the thalamus is a relay to all of those different types of sensation. And so that information will get relayed to the thalamus and ultimately to the cortex of the brain, to the hypothalamus and the limbic system for emotional regulation and the ability to kind of perceive that with the emotional ability, and then the parasympathetic activity as well.